what we are doing. I am French, sorry for the accent. Hope you will manage. The presentation will be slightly weird. I want to take you on the journey of the company, like why we have made what we have made and why we are where we are at the moment, and which is the thought process that actually leads to the collaboration with Atelerix. So as all company, we start with a problem. What is the problem? I don't know if you're familiar with the drug development field and the pharmaceutical industry, but usually at that moment, we are 70 times less efficient in producing new drugs per billions dollars invested. It takes longer, it costs more money, and we're producing less drugs. So they might have at some point a problem. When you dig in, it happened that more failure are happening during clinical trials, and particularly in the phase 2. At the moment, you are actually evaluating the efficacy of your molecules. So if you take it a bit backward, if it fails on the phase 2, it means that it has already passed many, many, many tests. People have taken the decision of pushing toward clinical trials or human testing, and yet again, it fails, meaning that probably the tests that has been made before were not super relevant or translatable to what is observed in the humans. And of course, what test are we talking about? The classic 2D cells in a dish and the animal models. So meaning that probably these specific methods are not adequate to the new drugs that we are developing. Globally, to put it a bit in numbers, in the drug development phases, we are talking about 90% average failing rates, meaning that if you want to be a bit sarcastic, flipping a coin is 50-50, you would be more predictable. You can try to defend this toward the FDA, good luck, they will never accept it, but on the statistical point of view, it's working. And when you have immune cells involved, these 90% are actually increasing to 95%. So you are absolutely not predicting, you are not understanding what is happening in human. And it's the same when a drug is already on the market. We are using it, particularly in oncology. And overall, 30% of the cancer all indication will make a relapse, meaning that we're also unable to predict which cancer will actually have more aggressiveness and adapt the treatment to it. So is there a solution? A priori, yes, and otherwise I won't be here. So one of the ideas is developing preclinical, so in vitro models that are closer to the human physiology. We are not 70 kilogram mice, we are made of 3Ds. So there is a field that have now almost 2 decades. It started with organoid chip, 3D cell culture, spheroid, organoid, and now every oid you can imagine. You have assembloid, organismoid, etc. Overall is building up 3D structure that recapitulate as best as possible an organ function or a pathological function. The field started in academia. It has been pushed a lot. There is beautiful papers, beautiful proof of concept, and since 5 to 6 years, it's also heavily pushed by regulation, particularly in the US. The FDA has actually lifted the mandatory animal testing. At the moment, you claim actually a first in human trials. However, it's super nice on the paper, but when you look at the data, it's absolutely not adopted in the daily practice of our beloved pharmaceutical industry and biotech in general. So we actually ask ourselves, why? And by conducting many interviews, many discussions, many field trips, we actually have ended up identifying 5 big problems that you can group in the scientific bottlenecks and the industrial bottlenecks. For the sake of these talks, I will just summarize by saying in the scientific bottlenecks, people need more complexity. In the industrial bottlenecks, they want something that is user-friendly and they can actually have access to super quickly, everywhere. The everywhere is actually the reason I'm here today. Before going a bit more in details, there is a few concepts that I would like to explain and to pass. The first one is the fit for purpose. Why it's important? Because actually when we are developing a model, it has to fit something. It has to answer the specific questions. So if we take an example of what it is, if you want to move from here to the Cambridge campus, you will probably use public transportation, bike, cars, etc., or scooters. If you want to move from Cambridge to, let's say, Mumbai, India, You will use another way of transportation, probably plane. You can try by car, good luck, but why not? You can try ships, etc. Then if you want to get to the moon, you only have the rocket options, but you have different design of rocket, okay? So the means of transportation is actually fitting your goal, okay? And the distance you have to cover in this case. So why is it relevant in drug development and in human physiology, and particularly in 3D models? We want to recapitulate organs' functions. It's a bit impossible to recapitulate all the organ functions. The talk before for the hepatocyte was particularly striking for this. Hepatocytes and liver is a myriad of functions. Having a single model that is highly performing in all these functions is first complex and more likely impossible to obtain. So we need to select based on the questions we are asking. This is the fit for purpose. What do I need to answer these questions? A second concept is the technology readiness level. All good story starts with a rocket story. This one starts in the NASA when the president of the US say, what if we put humans in the moon? So you have to coordinate a lot of people, and they all have to agree when they say, we are ready. So they actually build these scales. You have two main thresholds. The first one is 3. We have a prototype that is working. It can fail 40 times, but if it works 1 or 2 times, it's a proof of concept, it's good enough. Not at all ready, but we have some good clue that it will be working. Then you have TRL 5-6, which is a working prototype in an environment that is close to the one that will be the final one. And for everything that is above, it's industrialization, worldwide shipping, and standardization and robustness. With all this in mind, I hope I haven't lost you too much. Our idea of the solution for the adoption of 3D cell culture in general was to have a platform, hardware, that will actually standardize the culture, standardize the protocol, having a specific organ set that we know exactly where it's working well and where not to use it, and all this for specific models that are usable everywhere, so above the TRL 6. How does it look? Like this. We have the hardware that is here, we call it the cubics. We were not genius in marketing, it's cube, so it was quite straightforward. I can tell you more detail on the genesis of the name, but for the sake of this presentation, it's called the cubics. It monitors and controls the experimental conditions, pH, temperature, flow rate, the eventually stiffness of the matrix, the level of shear stress, and so on and so on. So you can actually tune if you want bone, if you want brain, if you want liver, if you want muscles, or pancreas, for example. Then, because it has to be used by the industries, we need to fit on a standard. So we fit on the standard multi-well plate. We do not use at all microfluidic chip, etc. So we get on the Falcon, etc. Standard multi-well plate. This is what you see here. We had developed actually an adapter that will control the biological conditions at the closest to the biological culture. And because we are operating worldwide, or we aimed at operating worldwide, we also implement a database of protocols, because we can develop something in Cambridge, we can develop another protocol in Paris, but maybe someone in Sydney or in Stockholm would like to have the same questions and would like to use the same protocol. So with the system, you just learn, it's a bit like a food processor, you follow the recipe step by step. The recipe and the protocol being attached or developed on the hardware, you will have constant and a standardization on the culture condition between Sydney, Stockholm, Paris, and Cambridge. It's private or public, depending on what you want and the use. So with all this, we end up today by having developed these 3 main axes in the 3D cell culture or organoid culture, either from the simplest protocol here on the left to the more complex one where we have biological interface, where we have dynamic system, eventually immune cell circulation, etc., etc. Just to showcase a little bit before getting in the collaboration with Atelerix, the use case. So the hypothesis we had at the beginning is we need to sustain more complex model to recapitulate specific function of an organ or a disease. So the very first area we operate was the metabolic diseases. So we take adipose tissue. It's fairly easy to actually isolate stem cells from the adipose tissue, from fresh adipose tissue. So we make the comparison. On the left is in an incubator in a complete static situations. On the right is actually what we have developed using the controlled cubic system. On these slides, before getting a bit more in details, what you can observe first is after roughly 20 days of culture, you have necrotic core happening in these organoids. The green are the lipids, so the fat that are on the cells. And in pink, you have actually the endothelial cells, so the blood vessel cells. And with time, you're actually completely losing these blood vessel cells, which is not what we can observe on the right parts. To get slightly more in details, you should imagine that on the top here, this is where we are the perfusion, so the bloodstream-like culture medium with oxygens, with nutrients, etc. So you can observe that the endothelial cells, the blood vessels, are actually differentiated from stem cells towards the oxygen. They are looking for oxygen, so they are going toward it. On the other hand, which is exactly what is happening in your human body, the adipocytes, so in green, are actually differentiating against the gradient. They are actually getting away from the oxygen while differentiating. So you have non-mature cells here and much more mature cells at the bottom. As it's what we observed in the fresh tissue, we were super happy. We can maintain complex biology starting from stem cells. Perfect. Second question, is it relevant? Because this is the hypothesis. Complex will means clinical matching. So we moved to the breast cancer. And we, if you read the slides from top to bottom, it's different level of complexity. Tumor cells alone, two type of a co-culture of two type of cells, and then the full microenvironment of the tumor cells. The only important things here is the yellow one. Yellow means cancer. So you can observe that what is normally used, you have only your tumor cells. In our model, you cannot even say that it's actually a cancer. If you use it without knowing, it can be a fibroblast or whatever other type of cells. And we actually restore this expression of the tumor marker with a complex microenvironment. And something that is even more striking, we had access to the history, the treatment history of these patients, but not the results of the treatment. So blindly tried it. This is after 15 days of treatment. We observed first a decrease in the yellow marker, so less cancer cell. But after 15 days, there were some spots that were becoming more and more important of new tumor cell. So we get back to the clinician saying, seems to have a problem. And actually what we didn't know is that this specific patient had a very aggressive cancer that make a relapse and become completely resistant to the tumor she was, to the treatment she was undergoing. So we had an in vitro system with enough complexity that actually recapitulate the clinical outcomes of this breast cancer is actually relapsing and becoming resistant to the treatment. The huge added value is we could actually explain why, which is not possible when you are actually making an MRI, etc. Because the system can stay on a microscope, we isolate the clusters that were actually co-expressing the basal, so normal marker, and the tumor marker. And when you killed the tumor cells around, they actually flipped and become cancerous while they were considered normal before. So that was, for us, the first ultimate proof that the organoids were able to manufacture in-house with a certain level of complexity is good enough to be used a bit widely in order to predict what will happen when you develop a drug or when you try to actually establish a strategic treatment for a specific cancer. So what is now and next? We actually propose this expertise on this level of complexity worldwide. So meaning that we need to actually ship this type of plates loaded with organoids. This is what we call the organoid plate. We had developed and offered some off-the-shelf model, the breast cancer, the adipose tissue, etc., that we characterize every time. But the know-how is also being able to make custom organoids, either primary, it can be differentiated from iPS, it can be primary, immortalized cell line, etc. What I want to focus right now, because this is what we have validated with Atelerix, is on the adipose tissue one and on the breast cancer one. just for the color code, because it will be the same for all the next slides. Green is fat, pink is blood vessels, and the red are actually fibroblasts. So what you can see here is we're also able to monitor almost automatically the growth of the different organoids. So this type of plot that you have here is actually the size distribution of all the organoids in one well or in all the plates that we're able to monitor during the time. So if we start with the adipoplates, we have a huge amount of constraint in shipping adipose tissue. They cannot be frozen. And the talk of Steve earlier, we had actually this misfortune where we shipped a plate in the US right after the UPS plane crashed. It was in Memphis, I believe. So all the logistic was disrupted, and by habit, they placed the parcel at 4 degrees, which was absolutely dramatic for the culture. So they killed everything. So adipose tissue cannot be frozen. Other solutions, so cryopreserved is ruled out. And there is this portable incubator. They are cheap. They are a logistical nightmare to collect back. So it wasn't possible. And actually, Atelerix strike, I don't exactly remember how we found you, but probably online first. And so we decided to try. Is it really possible to actually ship at room temperature? Honestly, as a scientist, I would never, ever have imagined this possible. So I had my own doubts, same for the team. So as mentioned before, we tried ourselves. And actually it worked. So this is a time course. You have here the first induction media, so just to start the differentiation of the stem cells into the fat tissue. Once it's done here, you have a normal maintenance medium that we did in our own setup, and then we have 4 days in Atelerix. 4 days is usually the time for the shipping. We have pushed it at 12 days. The results are quite similar. And at the moment you are actually removing them from the hydrogels, you see that the organoids continue to proliferate. This is on the size. They are also completely metabolically active in terms of their answer for beta-adrenergic stimuli and lipid content. Just for the sake of the presentation, you have an image here at the beginning, and you see that actually at the end you still have these beautiful roundish smooth cells. All these little brighter areas that you can see here are actually the lipid droplets that you can see through using the microscope. We did, of course, the same type of study on the breast cancer. In this specific model, the constraint is maintaining a highly complex model where we have 7 cell types. At the moment, we are actually building it and before shipping. So we have the cancer cell, but all the microenvironment with a really high biomass, meaning that in a transport that will be more than 24 to 48 hours in normal conditions, everything would die, or you start to signal actually apoptotic or necrotic cells. So the model cannot be used at all. In this case, if we still follow the time course, it was a 5 days shipping time frame that we took. This is how one well is looking. So every green dot is actually one organoid, and you can see that before or after the shipping, it did not affect at all the viability. The organoids are still there and they can still be used. We have pushed the specific breast cancer model until 36 days, I think, of the after Atelerix. And honestly, we cannot see any differences in viability, in reactivity to drugs, etc. With that, I would like to thank you for your attention. I hope it wasn't too boring as the last speaker, and I would be happy to take any questions.